So up to now we have discussed only about implicit solvation. Now there are explicit solvation also, and the explicit solvation is required when the explicit interaction of the solute with the environment is important. One of the examples is electron affinity of uracil in water, and we are talking of the adiabatic electron affinity. That means the energy difference between uracil, neutral uracil at neutral geometry and anionic uracil at anionic geometry. So this is how the electron attachment to uracil looks like. So initially the electron is delocalized over the water molecule, so it's like a pre-solvated electron, and then it's eventually get transferred into a valence bound uracil state, and between the neutral and the anion the structural difference is quite high. Now in this case, because I am dealing with an anion, there is a very strong hydrogen bonding of the anion with the surrounding water molecule. Not in the case of uracil, but in cases of certain nucleobases, there can be a proton transfer. Proton transfer from water is very unlikely, but if you have any other solvents, like if you have amino acids surrounding the nucleobases, there can be a proton transfer from the amino acid to the nucleobase. So how one model the solvent distribution around the solute is very crucial in these cases, and because there is an explicit interaction, sometimes proton transfer, this cannot be treated by implicit description. You need to treat the solvent explicitly, but problem is that what we have discussed before also, that the solvent distribution around around any solute in a real life situation is dynamic. So it's changing with time, and what you see in the experiment is a time average of it. So how one model the solvent distribution around the solute is actually crucial. And generally the way it's modeled is generally one takes snapshot from MD run. So you run a classical MD simulations over a long time and take snapshot from different parts of the trajectory, and then do the calculation and you average over it. You see that the averaging is going properly, that your results are converging with respect to averaging or not. And of course, if you have such a big system, generally a QMM method is used. You cannot do a QM calculation on entire uracil water box. So I generally, if I take uracil water, then I will do QMM by using uracil as the QM and rest water molecules as MM. This is I am taking as a representative system. You could have done any other systems also. Now in this kind of calculations, when you do the explicit solvation, question is that of course the solvent is a bulk media, and does not matter how many water molecules I am taking, I am working with a finite box. So now question is that so that means I am supposed to take a large water box for doing my calculation. Now question is that how large is large, and when it can mimic the actual bulk water, it's very difficult to predict. Second thing is that most of the cases I am working with a non polarizable force field. So that means my MM region is polarizing my QM region, but QM region cannot back polarize the MM region. So that can lead to a problem. That's why how large I am taking the size of the QM region is actually important. And secondly, how I am treating the boundary. Boundary means here of course there is no explicit interaction between the nucleobase and the water. Sorry, of course here there is no bonded interaction between the nucleobase and the water, but when I say boundary means I am taking out the boundary of the box. So the water molecules which are at the boundaries sometimes behave slightly differently than the water molecules what you see inside the box. So how you treat these boundaries can sometimes leads to artifacts in your calculation that one need to take care properly by doing this. Now of course for this kind of system you have to use certain protocols for doing calculation, and this is the detailed protocol. For the problem with explicit solvation is that for each kind of property, for each kind of system, you have to properly benchmark the protocol. So one protocol you cannot use everywhere, and that's a big disadvantage compared to the implicit solvation model, where you actually can just tell the solvation model and solvent name and can do the calculation. So the protocol what was used for this kind of system for electron attachment to the nucleobases, that you first run classical MD of with uracil water for 10 nanosecond and then generate 10 snapshots. You run QMM MD with each of these snapshot for 2 picosecond, and from each of the snapshot you get 400 snapshot out of it. Calculate the vertical detachment energy, and from that vertical detachment energy you do some pre-screening. Then from that pre-screen snapshot you optimize the neutral and anion. And remember, we have shown this method LOPT, because if you have to optimize such a big number of molecules, you cannot do it with a normal optimization scheme. So you optimize them and then you do the single point calculation using EAUM CCSD. You check the convergence of the result with respect to the basis, because I'm talking with respect to the I'm talking of electron affinity which converges very slowly with respect to the basis, especially if I am using a wave function based method. And for the other snapshot also, I need to check the convergence of the electron affinity value with respect to the size of the QM region, convergence with respect to the water shell size. So basically that helps you. So you say first shell of water, second shell of water, third shell of water, and that helps you to and extrapolate it to infinite number of cells. That helps you to remove the boundary artifacts and convergence of the of course, you have to also check the convergence with respect to the basis set, and all of them actually goes into the final adiabatic electron affinity value, and the final calculated value reported in this paper is 3.24 and experimental estimates is 2.94 to 3.40. So these experimental estimations are actually calculated from their major reduction potential. So the thing is that you can see to do use explicit solvation is a complicated process, and it requires a lot of steps, and you really need to be an expert of this kind of solvations. And to do this kind of calculation, you need to be good with molecular dynamics, QMM simulation, sampling, all sorts of things. So for most of the cases, these are not advisable. And the question is that, okay, if I use implicit solvation, in this case, how bad will it look like? So let us do this calculation quickly. So let us take an uracil. Maybe Chemcraft will be a better solution to get nucleobases. Add fragments. Molecules, DNA, uracil, I save structure with simple XYZ format. Let me make a new folder called uracil. So let me optimize it, maybe with PBE D3 opt fric. I want to use RI, and because I am using an ion, maybe we should use MADF2 SVP. Let's first optimize the gas phase, neutral, and then we can go to the solvent phase. U neutral gas phase. Orca Vodka. So the neutral calculation has been done. So we, for the gas phase, of course we can do the anion calculation. And we can just use the XYZ file from Uracil. I have to make it an ion, I need to check the charge and multiplicity also. So the name of the file has been wrong. This has to be U.